Juste une minute, donc je suis content de présenter Monsieur Reto Brun, qui vient de partir à la retraite, qui était à Bâle et qui a beaucoup travaillé sur la mise au point de drogues pour le traitement des protoses, en particulier le paludisme. Donc il fait partie des gens que je ne connaissais absolument pas, partie des gens que je sélectionnais sur le fait qu'ils étaient highly cited, qu'ils avaient trouvé des choses qui sortaient de l'ordinaire et qui faisaient qu'ils étaient parmi les gens, pendant une période donnée, parmi les plus cités. Donc, comme je ne veux pas négliger la parasitologie, donc je lui ai demandé de venir et il a eu la gentillesse de venir ici, bien que malheureusement il n'y ait plus d'avion direct Bâle-Marseille. Thank you for coming.
Thank you. So, please forgive me for not speaking French. Yeah? As a Swiss, I should be able to speak French. I understand it. I speak a little bit, of course, but it's much easier in English. Okay? Thank you. Well, I first got in touch with tropical diseases about 45 years ago. That was in Uganda and in Tanzania, where I was participating, had the chance to participate in surveys on African sleeping sickness. So African sleeping sickness was my first disease I got in touch with, and I worked on this disease the whole rest of my life. But of course, I also did other things. Other diseases that joined, like malaria. And while my research focus was on the control of the disease, later on it shifted to drug discovery. And for the last 25 years, I was mainly doing drug discovery for human African trypanosomiasis, or sleeping sickness, and for malaria. And I will also cover this in my presentation. Now, neglected tropical diseases, is actually a rather new term. We used to talk about tropical diseases, and then all of a sudden this neglected came up. And this is very, very important, because there's a number of diseases which does not catch any attention, and these are those neglected diseases. And also a new term is elimination. This is rather new. We always talked about we have to control this disease, control. That's all you can do. You cannot go beyond. But now, since about 10 years, we start to talk and also to believe in elimination. And elimination, you know, is the first step of eradication. But that's a much heavier business. So, neglected diseases is a group of diseases defined by the World Health Organization. You probably all know them. They belong to 4 groups of pathogens. Protozoa, we have Chagas disease, sleeping sickness, leishmaniasis. They're also called most neglected. And malaria, which does not belong to these neglected diseases, but I always put it on the list. Why? Malaria is the most important parasitic disease to begin with, and in many, many parts of Africa, malaria is still a neglected disease. Then we have a whole list of helminths. These are diseases caused by all kinds of different worms. Very funny creatures among them. And then we have a list of bacteria: buruli, leprosy, trachoma, yaws, mycetoma. Mycetoma is caused by bacteria and by fungi. It's a new interesting disease. And finally, 3 viruses. Maybe Zika should also be on this list, but it doesn't matter. The impact of these neglected tropical diseases is very significant. About 2.5 billion people are affected by it, and the mortality if we include malaria, is just about 1 million people dying each year from these diseases. Now, when we look at the global burden of these diseases among the infectious diseases, then this any statistics is dominated by the big three: malaria, AIDS, and tuberculosis. They cause annually about 200 million DALYs. These are these life years which are lost due to death or disability. Now, when we look at the neglected diseases one by one, then you see that each disease is not contributing that much in terms of DALYs. But when we add them all up, then we get a sum of 26 million DALYs, and this is a significant number. So we cannot just ignore neglected tropical diseases. Now I want to discuss a little bit factors which influence these parasitic diseases. First of all, we have a group which we can summarize as human behavior. We have migration, which plays a role, and in red you see examples of diseases where you have this connection. Then we have civil war and civil unrest. We have change of land use, change of food supply. An example here is, for instance, the introduction of aquaculture in Asian countries, and with aquaculture you have a proportional increase in foodborne trematodes. Then, of course, climate change. I'm not going to talk about this, but it's quite obvious that climate change, global warming, leads to an increase in vectors. This is not more than logical, but it's not only the vectors which increase. It's also that the parasites, they develop faster in the vectors. And also we have to think about flooding, for instance. And flooding creates new breeding sites for insects, but also living areas for helminths. So this is a huge area, and the influence is enormous. Not only in a negative way, also some of the parasites, they also get problems when the temperature is rising by 2 or 3 degrees. Then, of course, control measures, they influence the outcome of parasitic diseases. Here we have vector control and drugs. And I just talked a few minutes ago with Professor Raoult about vaccines, We don't know what the influence is because there are no vaccines for these diseases. Then we have co-infections, which are important. And here I think especially about HIV, which positively correlates with TB and with leishmaniasis in particular. And then at the end of my talk, I will talk about international solidarity. This also means campaigns to eliminate certain diseases. Okay, an interesting example of migration is Chagas disease. And Chagas disease is actually restricted to South and Central America. But we have many migrants these days, all over in Europe, in the capitals especially. And a study by François Chapuis in 2008 was quite amazing. It showed that 13% of the migrants they examined were seropositive, which means that they are carrying the parasite. They were not sick, but they were carriers of the parasite. And potentially they could transmit this disease. Now, fortunately, we don't have the vector in Switzerland, and not the vector not in the whole of Europe. But Chagas can be transmitted by blood transfusions, by organ transplantation. And also from the mother to the child. So it's not a catastrophe, but we have to keep that in mind. Influence of civil unrest and war. A very good example is African sleeping sickness. You remember, this is a disease I really like and I worked on the whole of my life. Now, the blue bars, they show the cases in Central Africa. And you see that the cases, they came down to the 60s, and then they were on a rather low niveau. And this was because of the colonial forces. They introduced rigorous control measures, and they were able to keep the sleeping sickness under control at a level which was hardly detectable, you know. But then at the end of the 60s, all of these African countries, they gained independence. And then what happened, you know, control measures were neglected. And some countries, they had civil wars. And so the cases started to go up. And by the end of the last century, we reached almost the same level. You know, it was really alarming. And then something happened. You know, control measures were introduced again by WHO, by other organizations, and the cases came down again. Very interesting. So this is a disease which can really well be controlled. Now, let's look at Angola. Angola, 1974, had 3 reported cases. Then the country became independent, and the cases started to shoot up. This is just an estimate of suspected cases in the year 2000. And then again, control measures were introduced by different organizations, and the cases came down again. Now, control measures in general, what possibilities do we have? We can survey a population with the aim to isolate and treat infected individuals. We can do vector control. This is usually very effective. Drugs, the use of drugs is always helpful. We can think about mass treatment for some diseases. This is very effective. And for zoonoses, these are diseases, you know, where animals and humans are affected. We can also think about controlling the reservoir. Now, let's go back a little bit. This is a very old picture of, it's taken in Cameroon around 1930. The French army was there, and the French army was aware of sleeping sickness. And they were really implementing good control. So what happens here, you know, the whole village is being invited to appear on the central place for examination. And here we have a whole battery of microscopists. Each person had to give a blood sample. These are the people, you know. This is the microscopist. He looks at his blood sample. And so the whole village was screened. If an infected individual was found, the person was isolated for treatment. Treatment by that time was a nightmare. It was by an organo-arsenic drug called atoxyl, terribly toxic. leading by itself to mortalities, yeah. Well, it was the only thing they had, you know. Yeah, this is really a fantastic picture. Here, this person, that's Dr. Jameau. He was in charge of these whole activities. Okay, when we look at this picture about 80 years later, this is also screening how it's being done today. Then somehow it's very similar. Similar in the sense that we still have microscopes and blood samples, but also very different. First of all, not the whole village is showing up, just the people who want to show up. So it's not compulsory. People are asked if they want to have their blood tested. Then this is a serological test which can be used in the field, which is being applied. Unfortunately, what you see here, this is a lumbar puncture. This still has to be done, or had to be done, for the typing to see whether the parasites are already in the CSF or not, because that was influencing the therapy. But this may change very soon, because we are soon having a drug which will reach the CSF. And then this staging is not important anymore. Now, vector control was and still is a very effective way to reduce the number of vectors. Insecticides, they were used in the early days. They were sprayed from airplanes, you know, which was not so good for the environment. But it was effective. We also have to mention that. Today we have insecticides which are presumably not toxic anymore, so-called pyrethroids. They are still being sprayed, maybe not from airplanes, but houses can be sprayed. But then traps play a big role, and here now I'm talking about vector control for sleeping sickness. The vector is the tsetse fly, and I spent a lot of time studying these tsetse flies and transmission and the whole thing. And you have different types of traps. On the left side, this is a so-called Bi-Conical Trap. This catches the insect alive. So if you want to examine the insect, for instance, want to dissect it, to find out whether it's carrying the parasites, the trypanosomes, then this is an excellent method. If you just want to reduce the number of insects, then you can also use a screen. This is simply cloth. Why is it black and blue? Simply because the tsetse flies, they like black and blue. Nobody knows why. You treat it with insecticides. The flies, they land on it, take it up through their feet, drop dead. Very effective. Now for mosquitoes, in principle, it is similar. So we also have, we can use insecticides. Indoor spraying is quite effective. If you do this twice a year, then this helps a lot. But then we also have to think about the breeding sites. We can treat breeding sites to reduce the number of insects, or reduce residual water sites, you know, and water sites, this is everything, yeah. It's just a flower pot or something like that, can be anything. And here, very important are insecticide-treated nets. And our institute has done lots of studies in Africa to distribute these nets, to also show the people how to maintain them. They have to be retreated with insecticides after a certain period of time. And they also studied the impact it had, and they could show that child mortality could be reduced by about 50% if you had a certain percentage of nets in the households. But of course, you still have ressentiments against the use of these nets. You know, some people they don't like to sleep below a net. But then this is then the role of the people, you know, to educate the population and to convince them that this is the way to really protect their children. Now, of course, we have drugs for these diseases, and now this is what I really like, you know. When we look at the existing drugs, or especially when we looked 20 years ago, then we mainly had old and toxic drugs. Terrible stuff, developed sometimes 50 years, sometimes 80 years ago. It was really almost a shame. Yeah, the number of drugs could have been limited, and still is, for schistosomiasis, for instance. It is mainly praziquantel, and the question is, what do you do if you meet resistance? In malaria, we have, of course, resistance problems with virtually any new drug, and also there are problems with the application and the availability of these products. Now, why was the situation, or is and was the situation so bad, you know? The big pharma, they stopped doing research and development for tropical diseases I would say in the 70s, 80s. The reason is very simple. It's too costly. And what they can expect back is not enough to cover the expenses. This makes sense, you know. A pharmaceutical company is not a philanthropic organization. They have to make profit, so that's why nobody was developing new drugs. And then, fortunately, something happened, and this was just about at the end of the last century, you know. It was WHO and the Médecins Sans Frontières, and they said, listen, it's too much. We cannot go ahead like that, you know. We are losing drugs almost every year. And there are diseases where we hardly have any drugs left. We have to find new ways. And then that was the starting point of these public-private partnerships, or product development partnerships. This is a joint venture of private and public organizations, also governmental organizations, big funding organizations like the Gates Foundation. And all of a sudden we had even many more than just the 10 you see here or so. Today we have about 40 or 50 of these organizations. They have the goal to produce products. It can be drugs, vaccines, or diagnostics for one disease, sometimes for several diseases. You know, they want to actually do the whole development with money from philanthropic organizations or from the public sector. And then, once they have a product, then they always find a company who is producing it for a cheap price. Yeah. Here I point out MMV, Medicines for Malaria venture, and DNDI. These are two organizations I know very well, and they also collaborated a lot. They are doing an excellent job, and you will hear more about it. But there are also organizations, they care for vaccines, and there is one even for diagnostics, FIND, in Geneva. I would like to show you a few examples, and also these are the good messages, you know, that we are going to have new drugs in the near future, in the very near future. And I start with new drugs for human African trypanosomiasis, or sleeping sickness. This is a project my team was also involved with DNDI. And the idea was to follow the strategy of the low-hanging fruit. And it was known that nitroimidazoles are very effective against bacteria, against protozoans. And the idea was to test all the existing nitroimidazoles on the market, or sitting around in some labs, you know. Some of them, they made it even in clinical studies, you know, but for others, there were at least in vitro and in vivo data available. And about 700 of these molecules were tested, and then a few dozens were tested in animal models. And after about 2 years only, fexinidazole was identified as a new candidate. This is fexinidazole. It is potentially mutagenic, but fexi is not really mutagenic. That was the fear about this whole class. A 10-day oral treatment, hundreds of patients could be cured in the meantime, and it's actually ready to be registered any day in Congo and in Uganda. It doesn't have to be approved necessarily by FDA, because it will never be used there. This disease is restricted to Central Africa. But this is really fantastic news. Now, fexi has an interesting metabolism. This is the mother molecule, which is a kind of a prodrug. But it shows activity in vitro by itself. So it's not 100% prodrug. It's rapidly metabolized to the sulfoxide and further to the sulfone. And both metabolites, they are active, even more active. Now, when we looked at the pharmacokinetics in mice, then it becomes obvious that the fexinidazole itself is being metabolized very rapidly, and the levels are extremely low, whereas the two metabolites, they show log 10 higher concentrations, and also a much more favorable. They stay for decent time periods at high levels. So this was in mice, and in humans it looks very similar. Now this is down here you see the fexi itself. This is the first metabolite, the second metabolite. This is a 14-day treatment with a very high dose of 3.6 grams per person. But the levels, especially of the sulfone, they are outrageously high. And this is really very, very good news. And based on that, the today's protocol is 1,800 milligrams per day for 4 days and 1,200 milligrams for another 6 days. And this cures patients, with so-called late-stage infection. This is the first oral treatment. All the other treatments, they had to be injected. And toxicity seems to be okay. Now, the NDI, they have a second project which was examining a new chemical class, the so-called benzoxaboroles. These are small, elegant molecules which contain a boron atom. And they also show excellent activities and also excellent pharmacokinetics. When you look at this here, these are plasma levels in different animals. For instance, the blue one is in a dog. They stay for several days. They hardly go down. Also, this is a monkey. So based on that and confirmed in humans, we can assume that a single tablet will cure a sleeping sickness patient. And this would be a revolution, of course. This would be fantastic. Now, this project is about 2 years behind the fexinidazole, so it will take another 2 or 3 years before this can be seen in patients. Now a quick look at new developments in malaria. You know that the latest generation of anti-malarials are the Artemisinins. They are extremely active, very fast acting, but they have, unfortunately, a very short half-life. Another drawback is the complex chemical structure, which you see over here. You cannot synthesize that. I mean, you can, but it's too expensive. So you have to extract it from the plant. Fortunately, the plant has a very high content. But soon the idea came up, we should try to produce a synthetic artemisinin. Now, the artemisinin, the pharmacophore, it was found out, is this peroxide structure here. So the idea was to go for a simple peroxide, here a trioxolane, stabilize it by two groups, left and right, and if it works, then we would have a solution, you know, because such molecules, they are easy to synthesize. We were also involved in this project. It was a slow process. It took about 20 years, yeah, but this was academic, you know. And here you see that is the end molecule, number 439. We started with number 1. And this molecule is now being produced by Sanofi. And it is directly being used in combination with ferroquine and piperacin, because in malaria you have to use combination therapies. So these products, they are not on the market yet, but they should be out also anytime. Another chemical class, a new one, are the spiroinolones. You see the molecule here. This was a project with the Novartis Institute for Tropical Diseases. A library of natural compounds was screened, and a molecule was found, a very promising molecule, which was not a natural product, but very closely related. And then, of course, it required the lead optimization, and this is the final product, this molecule. What you see here is results from clinical trials, and they show that it is very effective in T. vivax as well as in P. falciparum. And it's very fast. It clears the bloodstream within 8 to 12 hours, and here also 8 to 16 hours. This is very fast. And this is being developed now by Novartis. Okay, now we have talked about control, and I would like to move on and pose the question: will it be possible to eliminate some of these diseases, or even eradicate? You know the difference between elimination and eradication. Okay? Yeah? No? Okay. Eradication means the agent is gone, the disease is gone, and the pathogen is gone. This is like smallpox, yeah? That's the only disease which was eradicated. Now, elimination would be a step before, you know. It just means the disease is not a public health problem anymore. The prevalences are below certain levels. Yes, that's it. So it's something in between, you know. Now the London Declaration, this is a fantastic step. Just 6 years ago, you know. 6 years ago in London, this declaration was signed by all the major drug companies, by affected countries, by governments, by philanthropic organizations. And the aim is to eradicate the Guinea worm disease, to get rid of it for good, to eliminate these 4 diseases, and to control these 5 diseases by the year 2020. In the meantime, they also talk about 2030. That's not so important. Okay, now what about the eradication of the Guinea worm? The Guinea worm or disease or dracunculiasis was very predominant just 30 years ago. In 1986, it was assumed that there are still 3.5 million patients in over 20 countries in Asia. Now the last figure, and you can read that almost every day. There's a page where you can see how many patients they still have. So the latest I found is that they assume that there are only 11 patients left on this planet in southern Sudan and in Chad. And the whole thing is pushed by the WHO and by the Carter Foundation. Here you see a picture with Jimmy Carter talking to a village where education is being done. Education here is extremely important to tell people how they get infected and what they can do against it. And this is the pathogen, the Guinea worm. You know, it's a long worm living below the skin. It's not fatal by itself, but it can lead to infections, of course. It's a long worm, 1 meter long, and the female shows up, you know, in order to put out eggs, and then you have to catch it, and then you can pull, but just a little bit, otherwise it breaks. And then the worm dies, and that leads to heavy infections. So that's why you usually roll it on a stick, and you have to do it for several days. But the whole thing is extremely painful. It's not so much life-threatening, but it's disabilitating, and you cannot work with that, especially not when you have it in the foot. Now, another disease which is on the radar of elimination is sleeping sickness, African sleeping sickness. And when you look at the numbers of patients, yeah, in red you have the real figures, the reported cases, and then you also have the estimated cases. Why such a discrepancy? Simply because these are difficult countries, you know, and maybe only a small proportion is really under control. That's why we have these discrepancies. But now when you look at the figures, then you see that the numbers came down tremendously. This was certainly based on interventions, but maybe also on environmental changes, other things we do not know yet. Yeah. The latest figure we have is 2,800 reported cases, but the number must be higher, probably 15,000, but not much more. Now, elimination would mean that we have to bring down the number below 2,000, and also the prevalences in certain areas below certain figures. Yeah. Now, everything looks extremely good for this disease. Everybody is optimistic. It will be very costly. These are estimations based on modeling. The point here is that you have to do it for a long time. You know, once you reach this elimination situation, you have to monitor it for at least another 20 years. You know, and even then, if you stop doing it, what happens? You know, does it come back? Maybe, we don't know. That's why eradication would be so ideal, and also on the long run the cheapest method, yeah, because it never comes back. Now, this is something which made me a bit nervous. Yeah, this is a very recent publication from this year by Philippe Bücher's group. Okay, you have up here, you have the part in the animal. You have the animals, they are susceptible, they get infected by the fly. They are infective, that means they are also capable to infect or to allow new flies to get infected on them. We don't know much about self-cure. Of course, you also have treatment. So it's very complicated, you know. Now in humans, it is similar, but even more complex. We have susceptible people, they get infected. They go through a latent phase where we do not know that they are infected. Then they start to develop the disease. If they are treated, they revert back to susceptible. They can be reinfected, no problem. But what's important is this latent stage. Yeah? We don't know how important that is. How many people are out there, you know? We have these few thousand patients, but maybe we have 10,000 of latent people. We have no idea, because it's very hard to find them. Then there was a year before, there was another publication by British scientists that was pure speculative, but also it was very frightening. We know that occasionally a child can get the infection directly from an infected mother. We have no idea how frequent that is. So far, we assume that it's negligible. Yeah, but they now speculate that this is happening much more often, and this would not only mean that again we have this would feed this pool of latent people, but this also means that this mode of transmission does not require the insect, the tsetse fly, you know, and we are so hard chasing these tsetse flies. So even without tsetse flies, you know, this could work. So we could have a large number of latent people just waiting, you know, for a situation where they convert. For instance, it can be a war, it can be stress or something like that, and all of a sudden, you know, we get large numbers of patients. So this is something which could actually interfere with this elimination. Elimination is also on the radar for malaria. It is not so. It is rather new. In the year 2015, by the World Health Assembly, they also defined goals, and the goals are to reduce the number of cases and also the mortality by 90%. Then to eliminate malaria in certain countries. This already took place, and it's possible to do that. But currently, malaria has almost a worldwide distribution, you know. And then also this point 4 is important, you know, that once you have eliminated it from a certain area, you have to make sure it doesn't come back, because that's very easy, you know. Infected people travel. Also, the insect can travel, but they are good. There's good progress on site. This is something the Gates Foundation really put on their flag. They say we want to eliminate malaria. They don't give a year. Yeah, I think it's not impossible, but this is more something like for the next 50 or maybe even more years or 100 years. Yeah, but on the other hand, it's also important to have goals. And let's try it, you know. This is my last slide. I wanted to finish with the view of two people who should know what they talk about. this was an editorial by Peter Ottes and Sarah Baksoy. They are the editors of PLOS, Neglected Tropical Diseases. And they say that they believe that these 3 diseases are heading towards elimination. They expect significant gains among these 6, including malaria, in certain areas, of course, only. And that's interesting. They also believe that currently we are losing the battle for dengue, other arboviruses, leishmaniasis, and Chagas disease.
And malaria.
And malaria. I accept that, yeah. I also accept it here. Okay, with this I would like to stop, and I thank you for your attention.
Well, malaria, I think it's one of the things is that one of the things that we have neglected in general is the role. We don't know the whole cycle, and specifically the role of the apes in maintaining the part of the bugs. So, except if you go from elimination to eradication, you need to kill the apes. That's or treat all the apes, which is all the difficult.
You can't kill them. In principle, it's not a zoonosis. It's being called an anthroponosis. But this is only true for maybe 99%, you know, and we also have a small animal reservoir.
Yeah, but I think in general we have not been able to really work. Well, there is, you remember these papers that show that falciparum is coming from Gorilla. Plasmodium. And we found leishmaniasis in Gorilla as well. So these have not been very well studied because there are only few access to this. And I see only for which reason, you know, apes will not be susceptible to our bugs. I think they're all susceptible to all our bugs. It's just a question of chance that they need it.
Or very slight modifications.
Yeah. So I think eradication would be really difficult.
Yeah, in the case of malaria, I would not think about eradication. No. For some viral diseases, why not?
Yeah. Well, viruses are much more
and also maybe
adapted to.
And also maybe some of the helminths, like the filaries. I think there it could be possible. Yeah. Because everything is much slower. The numbers are much lower. Also, malaria is. I mean, it's crazy. How many parasites it can be in one human? You know, it can be up to 10 to the 12. Yeah. That's why you then have so many mutations. Also, yeah. If you just have 10 worms in your body, then that's another issue.
And then the capability of changing and finding solution is huge. All right, it's tough work. You know, we've observed something I can't believe that, you know, when we in the village where we were working, we use the net, impregnated net, and first what we see is that some of the mosquitoes become resistant to insecticide, and then the second thing is that the mosquito change their dinner time.
Yeah.
They bite earlier and
they
later.
Shift to daytime meals.
So it's a war model that you made, you know.
Yeah, you ruined the model. Yeah.
They change their dinner time. That's it.
Yeah, it's the same if you think about eradicating an insect vector. Yeah, there's a big chance that other insects may take over this niche.
And even we find that the mosquitoes are biting donkeys, you know, when they don't get too much. If you get too much nets, they will feel more donkey. we were supposing that we were really anthropomorphic.
That's
so,
okay.
But they say from donkey.
That's
so
okay, that's okay from public health and public health point of view.
Yeah, but as soon as the net gets old, after 2 years, you know, they still come back from the donkey to the humans.
Ah, I
Just
see.
a question of
Yeah.
Just another question. The fact that some chemical compounds of plants have biological effect in disease, positive effect of disease treatments, do you think it says anything about potential reservoirs, or is it purely coincidence?
A pure reservoir in what sense?
Does it say anything about potential reservoirs? I mean, can the plant be reservoirs for some of those pathogens, or is it just a coincidence that some chemical compounds?
No, plants. There is no plant known acting as a reservoir. No, I don't think so. On the other hand, we also found anti-malarial molecules in marine organisms. Now, a marine organism never has to protect itself from malaria. So this is coincidence, yeah. Sometimes it's not coincidence. I mean, that plants, they have to protect themselves from insects, for instance, you know, and also from microorganisms. Yeah, roots, they are exposed to bacteria, to fungi, so that makes sense that they have antibacterial molecules, antifungals, but in a marine organism.
Is it Artemisinin, is it efficient against anything else that we know?
Yes, it was used against originally Artemisinin against I think diarrheal diseases. Yeah.
But in plants, that may.
Oh, in plants.
Is it fighting against any kind
of
infecting protozoan or arthropod or whatever?
I don't know.
Against schisto also. Artemisinin, I think it's also effective against schisto.
Schisto and.
But in plants, what is it?
Yeah. What?
How useful is it?
How useful is it for the plant? Yeah. I don't know. It is. It's interesting. You know, there are so many Artemisinin species, but only the Annua has the Artemisinin. Here in Europe, we also have lots of all over. Here on the compound, you have Artemisia somewhere, but they don't have the Artemisinin, so it's just this Annua. I don't know why. Yeah.
Might be a reason.
Yeah. Everything should have a reason. Yeah.
To persist at the minimum, not to create, but to persist.
Yeah, because they have a dry content of 1 or 2%. You know, so this enormous effort. That's expensive. Yeah.
Very expensive.
So why do they do that? I don't know.
At least they get a kind of plant parasite around that they eradicate this.
Yeah, it could protect them from an insect possibly who tries to eat it, or a parasite. Yeah.
I was wondering if it was active against fungi, because they are very big plant pathogens.
Yeah.
His father was trying to eradicate tuberculosis. He was the guy
he was working at the WHO. He was in charge
of this
eradication program.
Really?
Yeah.
Oh, fantastic. No, this is.
But he was optimistic. He believed he could do that in the 20th century.
Yes.
But this is just 20 years later.
Not
so bad.
Not so bad.
Well, in reality, these kind of enterprises are always taking more time. But this is a success story,
Yes,
I believe.
yes,
Yeah.
It's very interesting. I don't know if you've heard that now dogs can also have Guinea Worm. It was thought that it was only a human disease, and in Chad, the dog was contaminated.
I read this. This is very recent, and And I also, it reminded me of the sleeping sickness story, you know, that it looks so straightforward, and then all of a sudden you know that in dogs you also have the worm. And of course, you cannot control all these dogs, yeah.
Maybe there is a weak control because it's really a limited area on the child.
Is it? Okay.
But I hope so. That's why there are not so many. I mean, there's cases that continue to decrease.
Well, as long as they are going down, the cases in humans, this is a good indication.